Hello and welcome to another one at D's ADD videos. Today we're going to look at some basic underlying concepts dealing with enzymes, discuss some terminology you should be familiar with in order to be able to discuss enzymes proficiently. So what is an enzyme? Well, most enzymes are proteins. Sometimes we got RNAs that act like enzymes. What do? They catalyze metabolic reactions. And how do they do it? Mainly through kinetic control. By placing species close together that are going to be reacting, putting them in the right orientation. We're going to go into all this detail much later. So, the basic premise is that an enzyme and a substrate meet, and there's a specificity to the enzyme and the substrate that they fit inside each other like a lock and a key. Okay, let's make it a little bit more interesting shape. So this enzyme here will meet with the substrate and create what's called an enzyme substrate complex, and then give you an enzyme plus product. So let's do this. And I'll do that. So there's the enzyme substrate complex, and then since it's a product, let's do a different color. Okay. So some basic terminology you should be familiar with. The substrate is the thing that the enzymes acts upon, the target, the thing that will become a product, or sometimes there's not enough energy to switch it from substrate to product, so it bounces back to substrate from that enzyme substrate complex. Specificity has to do with the fact that the enzymes are designed structurally and molecularly to only recognize certain substrates. Otherwise, it'd be total pandemonium and everything would be catalyzing reactions for everything else. Cofactors. Cofactors are going to be non-protein, that's the key there, substances that require, or the enzymes require for proper function. I apologize, it's 2:30 in the morning and I've worked a full day. So, non-protein substances that enzymes require for proper function, metal ions, an example would be magnesium and DNA polymerase. Okay, DNA polymerase is an enzyme that helps replicate DNA, copy DNA, but without that magnesium, it's not going to be able to do it. Coenzymes are basically the same thing as cofactors, but instead of ions, metal ions, they are organic molecules. All right, so the enzyme needs the coenzyme for proper function. Next, we have prosthetic groups. A prosthetic group is basically whenever the coenzyme is really tightly bound to your enzyme, and they act as a unit basically. All right, and that unit is referred to as a holoenzyme. Whenever the holoenzyme is missing its prosthetic group, it's catalytically inactive. That means it's basically turned off, and it's referred to as a apoenzyme. All right, and these are just some basic terms that you should be familiar with if we're going to discuss kinetics, cooperativity, what else do we have? Inhibition. All these things have to do with enzymes. So this is laying down your foundation. You should check out the next video, which is going to be basic kinetics, Michaelis-Menten kinetics of enzymes. All right. I hope I've cleared up any doubts you might have over the basic understanding of enzymes? Thank you and have a nice day.